We'll save the applause to the end, okay? First of all, great turnout. This is amazing. So thank you all for coming and making the time to hear about Reef Life Survey. We're obviously passionate about it. Everyone here tonight that does Reef Life Survey, we're all volunteers. Some of us are scientists as well in our day jobs, but we have all manner of people that get involved in Reef Life Survey. And we give our time and come and basically gather data which scientists then use to draw conclusions about the marine environment. It's a great program to get involved in, but it only suits certain types of people that are sort of prepared to sort of dig into things and explore. So I'll talk a little bit more about Reef Life Survey first up, and then I'm going to talk about what we've found in Jervis Bay over the last 10 years. And there are people here who I'm sure can add richness to what I'm saying, people like Sue, who's been diving in Jervis Bay for, however many years, a lot longer than me. And Brian, who's been driving our boat, who knows the area really well. So I'm very encouraging anyone who wants to ask questions or get involved in the dialogue towards the end. Okay. So this photo here is an RLS diver doing a survey. It was like Plantation Point maybe about 4 or 5 years ago. It's a bit dark because we use strobes to sort of light things up. It was actually daytime, but it looks like nighttime. But there she is doing the survey. She's got her slate here and she's writing down what she's seeing. So that's what we look like when we're out there doing surveys. Now, Reef Life Survey is an Australian success story. About 12 years ago, scientists at the University of Tasmania asked themselves a bit of a question. It's very expensive to get data from underwater, okay? You've got to send teams out with dive boats and so on. So the question they asked themselves was, is it possible to find volunteers who could do as good a job as scientists at gathering data? And a lot of citizen science programs, the conventional wisdom is, oh no, you've got to simplify things for the general public. They found the opposite. They found that yes, it actually is possible to find people who are committed enough and throw themselves at it and really dig into the IDs and understanding all the fish species. And so this program, which started as a bit of a I wonder if it'll work, is now in 53 countries. We've identified nearly 5,000 species as part of the program. We've done over 13,000 surveys. Most of those surveys are in Australia because that's where it started, and the East Coast is very densely surveyed, but we also go all the way around Australia on a regular basis and overseas. So when we're out there, what do we do? It's very important for science that when you gather data, you do it in a standardised way. So, for example, if we just went out there and just wrote down what we saw, this dive was an hour, that dive was 20 minutes, this dive was at 5 metres, that dive was at 15 metres, we couldn't compare one site to the next. So what we do is we have a very standard method that we follow on every single site, and that allows us to draw conclusions about how this site compares to that, how this site has changed over the years, and how different things might affect the results. For example, does it make any difference if you're in a marine park or not? So the standardised method is we roll out a 50-metre tape, just like you get at the hardware store. We roll it out at a fixed depth, might be 5 metres or 10 metres, and we record that. And we go along that transect tape and we record 3 types of information. The first, let's see if I can get this to work. Can you see the little dot there? The first we call method 1. So method 1 is all fish species that we see in a 5 metre by 5 metre window. So we're swimming along, we've got this 5 metre by 5 metre window. Yep, okay, so there's this sort of wrasse and there's a grey nose shark, woohoo, that's exciting. And we're writing down all the fish species. We also write down other things that swim, like seals or turtles. So that's method 1. Method 2 is we then look more closely at the bottom, because when you're looking around like this, you're not looking at the little ledges and stuff, and you're not finding little gobies and so on. So method 2, we search a narrower band, once again, both sides of the tape, 50 metres, and we record mobile macro invertebrates, so invertebrates that move, sea stars, shells, seashells, crabs, and fish that hide, we call cryptic species. So that's method 2. And then method 3, we don't call that, we call it photoquadrats. We then swim along and take 20 photographs along the tape. And those 20 photographs then get processed later to draw conclusions about, for example, how much kelp cover is there, how much coral is there on this site. So those 3 pieces of information then go into a big global database and then can be accessed by the public, but also by scientists. That whole thing takes about an hour, okay? So a transect takes about an hour, which is basically a tank of air. So you might poke around for 10 minutes afterwards, find a few more things, but otherwise that's your dive. Now, normally we try to do a number of trips each year and come back to a place on a regular basis. We try to come to Jervis Bay every year about this time. We try to do Sydney about the same time. We do Port Stephens in May, etc. But this year we're doing a particularly big project called Lap of Oz. And the reason for that is, a bit over 10 years ago, they did a lap of Australia by beg, borrowing and whatever boats and getting around. And so there was a big effort to map the coastline's marine life 10 years ago. Fast forward to 2020 and we proposed to some philanthropic organisations, we'd like to do it again, but we need extra funding. And so the Ian Potter Foundation came to the party and has funded us. So that means that this year we're repeating that thing that happened 10 years ago. So we get a decade of change. And there aren't many data sets in the marine science world that span a decade. Okay, so that's pretty exciting. So here we are back doing the regular Jervis Bay surveys, but also doing Lap of Oz. And as part of Lap of Oz, they also said, well, we want you to also do some community presentations. So that's why we're all here tonight, thanks to this project. Now, before I get into what we've observed in Jervis Bay, I want to paint a sort of bigger picture. There are things happening in the world which clearly are also affecting Jervis Bay. And so let's just talk about a couple of big picture trends to give us some background. And this is research that's been done using Reef Life Survey data, but it's been done for, say, 1,600 or 800 or 1,000 sites all around the world. And this study here, which was published in Nature in 2014, concluded that fish biomass is down by about 2/3 on historical levels. So we've taken about 2/3 of the fish out of the sea. Okay? When you go further up the food chain, we like to take the big fish by preference, don't we? We don't like the little ones as much as the big ones. The further you go up, the more of an impact we're having. So when you get up to the level of sharks, it's something around 90, in the case of this paper, 93%. Now there are other studies that have concluded 90% or 88% or whatever, but we're around the 90 mark. So you've got 1 in 10 kilos of shark in the ocean compared to historical levels. Now in Australia, so it's global. In Australia, you might say, oh, hang on, we're different. Well, actually we're not. So this study here, which was published a couple of years ago, found that over the last 10 years in Australia, fish stocks have declined by 30%. Now this is in line with both fisheries catch data and Reef Life Survey data. They both show this. So that's the background. Now, is this happening in Jervis Bay as well? Well, let's have a look. So here's now spotlight on Jervis Bay. Do you like that little bit of animation there? It's impressive, wasn't it? Yeah. These little purple dots are our sites. Okay. Now we don't get to do them every year because there's too many to do in a trip, but they're all the sites that have been done over the course of Jervis Bay's. Well, actually, these are the priority sites. There are some more. There's one up here at Currarong, for example. These are the ones we try to do relatively frequently. Today we're out here. We did drum and drumsticks today. 2 dives, beautiful out there. And the biggest silver sweep I think I've ever seen. Nearly a foot long. In fact, they're so long, I thought it might have been a different species, because when they're little, they're sort of more rounded. But as they get bigger, they get longer. I think, is that actually a silver sweep? So really nice to see some big fish out there. Anyway, so they're the sites that we do, but let's overlay on that the marine park. Okay, so where is the marine park? Well, the answer is, well, the whole area is really marine park, but with marine parks, you have zones. So whilst we call it all marine park, the pink areas are quite different to the yellow. So the pink areas, you're not allowed to take things out or harm animals or plants, so they're sanctuary zone. whereas the yellow areas you are allowed to fish in. So even though the whole area is marine park, we do have very different rules as you go around. And as you might remember, a lot of our sites are around here. So a lot of our sites pick up, in particular, this sanctuary zone and this one here. We also pick up this bit here. Some of these others we don't pick up for a couple of reasons. One, we've got to get there regularly enough. So if it's an area that's outside, it's hard to get there often. Like drum and drumsticks, we haven't got to for 10 years. So even though it's out there, it's not one that we get to often. The other reason we don't pick up some of these other areas as much is we only survey hard surface. We survey reef. So coral reef and rocky reef. We don't survey sand. And that's because if you're going to do a good job of recording what lives on sand, you need different methods to what we use. So we constrain ourselves to rocky reef, and that's why even though this is quite a big sanctuary zone, we only do these couple of sites here in Hymes. There's 3 or 4 sites that we do in that area. So the majority of our attention in terms of sanctuary zones just happens to be in here because of its accessibility and rocky reef. Okay, so how many surveys have we done in Jervis Bay? What do you reckon? A few hundred? Okay, well, let's have a look. Since 2008, so here you've got Chris. Where are you, Chris? You probably don't want me to point you out, but there's Chris. 2 other divers who aren't with us on this trip, but Lou almost came to this trip, didn't quite make it. Since 2008, 64 divers have come here at one time or another. They're all volunteers. We've put in place 56 sites and we've done over 2,000 surveys. We've identified 379 fish species. That's a lot of fish species. Okay? 137 cryptic fish, the little ones that hide. Sometimes they're big as well, like wobbegong. They're cryptic because they camouflage. And 271 invertebrate species. So a lot of diversity there. So I'm going to start out by awarding some fish and invertebrates some prizes. Okay, we're going to give the prize to the most common fish. What do you reckon the most common fish would be? Come on, we're all locals. We would know, wouldn't we? Scad, blackfish, nardo. No one's got it yet. Sweet, no. Marias, no. Okay, so I'll give you a bit of a hint. The most common fish is one that you don't notice because it's pretty boring. Okay. So, the prize to the most common fish. So this is the fish that we record on the most transects. 90-something% of times we go out, we get this fish. The white ear palmer. You couldn't find a more ordinary fish, but it's a very successful fish. It's pretty much everywhere. Now, if you fish, as in line fishing, you may never have seen this fish because it doesn't eat the sort of bait that people will use for fishing. So we often have a skewed view of our marine environment because of the way we interact with it. So next time you go out for a snorkel, keep an eye out for these guys. When they're little, they get little blue colours through their face. They're quite beautiful actually when they're young, but then they turn this drab, ordinary. They get up to 15 centimetres, maybe 16. So we saw some today we thought might have been slightly bigger, but let's say 16 or so. Okay, the most abundant. Now, on a transect, you might get 5 or 10 of these. So they're not very abundant. There's not a lot of them. It's just there on every transect. The most abundant is the fish that we count the most quantity of. What's that going to be? NATO, scad. This one's flying under the radar again. It's the hula fish. It's about this big, and you get little clouds of them, particularly down in low areas. And I don't know, there's been thousands and thousands of those. I mean, we get a few hundred of those on every transect pretty much. But there are some transects that don't have these, that have these. That's why that's more common, that's more abundant. The most biomass. So now this is abundant but also big, right? This is abundant. They're only this big, so there's not much weight to them. What do you reckon? Someone has already said this one. The fish that is a big-ish fish that there's a lot of. Sue got it. It's the yellowtail scad. Okay, so there's more of these, but one of these is so small, whereas one of these weighs more, so the total biomass. So if you say the weight of fish in Jervis Bay, that's the winner. So they're the fish awards. Let's now give the Invertebrate Awards. Now, I'm putting a question mark on the Invertebrate Awards because the winner is both the most common and the most abundant. But maybe that's not a good thing. That's why I put a question mark. Which invertebrate, so crabs, sea stars, sea urchins, seashells, abalone, whatever, which invertebrate do you think is, now you're not allowed to say, Sue, because you will know this, the most common and the most abundant? We get 400 or 500 of these on a transect. Right, the long spine urchin. Now I'm saying question mark as to whether this is actually an award, because the downside of high abundance of urchins is they like to eat kelp. And kelp is the forest. So they clear the forest if they get a chance. So whilst that's good, because hey, we've got lots of invertebrates, they are an animal, it's nice to have them, the downside is that tends to correlate with less kelp, and kelp is good for other species. So for example, woody sea dragons need kelp. And so if the kelp goes, the Wheaties will go too. But the runner-up, because the black urchins is like what everything, I'm going to give a runner-up prize now. The runner-up, which is the second most abundant. Anyone want to have a stab at this? Sue? Yes. Sue's right, it's the tent shell. One that you probably don't even notice. They're a bit like those Palmer. They fly under the radar, but there are hundreds of them. They're a little cone, little pyramid sort of shell about that big. And we go out, when we run a transect, we count these, okay? Oh, you get better things to do with your Saturday, but still. I count them in tens or even hundreds. I don't actually count individual, otherwise I'd still be out there today. Okay, so they're the awards of the winners. Let's now talk about trends. What changes have we seen occur over the last 10 years? And some of the indices that we use aren't trending, so they're pretty well level pegging, so I'm not going to bother putting them up. I'm going to put up 3 indicators that are changing, and then we're going to actually look at some of the species that make up those indicators. So, trends, who knows where that is? Yeah, beautiful place. We've got a site at Currarong on that little bit of reef that's out there, but I think the creek's amazing. So here are the 3 indicators that I just wanted to show you. We do more indicators than this, but like I said, the other ones are pretty level, which actually isn't a bad thing, and I'll mention that in a minute. But this one here is big fish biomass, so it's a strong indicator of health of fish and the impact of fishing in particular, because we only count the biomass of fish that are bigger than 20 centimetres. So that means all those little hula fish and whatever are kicked out of the data set. So this is an important one because we care about the bigger fish often, depending on commercial factors and recreational factors. And the good news is that big fish biomass going up and down, your seasonal factors and recruitment of larvae and so on, but it's kicked up here. So this is the log scale. So that's gone from 1 to 1.5 on the log scale. That means that it's maybe 2 to 3 times that's gone up. Okay, so big fish biomass is improving in Jervis Bay. And that's a really good story. Okay. Now, here we've shown the partially protected areas, those yellow areas on the map, and it's green here, but it was pink on the map. These are the fully protected areas. Normally, we'd expect to see more separation between them, but here they tend to be tracking pretty closely, and it's nice to see that upward trend. There aren't many places where we're seeing that. So this is nice, the marine parks here, people clearly are, they care about this place, they're trying to look after it, and the fish population seems to be doing well. Fish diversity is also going up. So on average, back here in 2008, we were getting about 18 fish species on the survey. Now we're getting 23, 24. So that's gone up by about 30%. That's biodiversity, fish biodiversity. Another good indicator. And here are those cryptic species, all the little ones that hide, and they've also gone up even more. They've gone from, on average, 1. So you can see the actual data points. Sometimes we get 4 or 5, but 1 species on a transect, and now we're up around 3 or 4. So some good signs there in these indicators. Invertebrates, the indicators are all pretty level. Now, to me, that's still good, because the macro trend that we're seeing in the east and the south is invertebrates are declining in many areas. So the fact that they're level pegging here in Jervis Bay, to me, is also a good sign. So let's look at some of the species that make these up. I better hurry up, because otherwise I'm going to hold you up too much. So these are some of the more common ones. Now we have graphs for these. So you can see the Maori wrasse here. You can see it's going up, and you can see there's more of a sanctuary zone versus habitat protection area effect on these. Port Jackson sharks are doing okay in sanctuary zones, but they're declining outside of them. Now, in all of these graphs, I mean, I could have a stab at what I think might be going on, but I can't actually say. I can only tell you the trend, because RLS data tells us the trend. To understand why, you need to do more research. So I can speculate, and we can all speculate. Tonight's presentation is just showing you the actual trends. So you can see Port Jackson's aren't doing well except in sanctuary zones. Red Morwong, general uptick. Now, why is this one flatter than that one showing more of a curve? The way these models work is if they don't have a lot of data, they tend to be a flatter sort of picture. So the fact we've got a bit more of a pattern in the sanctuary zone means we've just got more data points, so it can pick up a curve, whereas the other one doesn't have as much data points, so it's a flatter. But they're both doing pretty well. Blue groper doing well as well. Now, one of the things that blue groper benefit from is they're protected from spearfishing in New South Wales. So there's a general thing happening that's helping them, and then there's also the marine park. So some good stories for fish. Invertebrates, you can see that maybe not so good. There are a couple that seem to be doing okay, but good old black urchins are kicking up or level pegging. The way you read these bands is if they overlap, you can't statistically separate them. So even though the green looks like it's going that and the brown, actually we can't say that, okay, because the bands aren't separated. If you go back here, we can say here because there's white space between them. So the bands are telling you whether you can actually read anything into those patterns. Here, level pegging as well on this, which is pretty good because these guys need kelp, or often are in kelp. So that sort of is a bit of a sign that maybe the kelp's sort of handling things okay. The firebrick sea star is trending up, and good old octopus. Who's been out to Octopolis? Got that sort of Octopus City area out near Murray's. It's amazing. I'll come to that in a minute, actually. I've got a picture. Cryptic fish. These are very common, very successful. The half-banded sea perch trending up. Unfortunately, moray eels seem to be coming down. Bullseyes seem to be coming up. So you can see there's a mixture of things going on with various species. Beardies, you can see maybe a bit of a potentially sanctuary zone versus habitat protection zone effect here because they are separating. Now in kelp, we don't have enough data to give you a trend, but I can tell you the actual percentages, how much kelp is out there. What do you reckon the percent of cover, if you took a photograph of the bottom and you said, you know, it's 20% this and 40% that, what percent of kelp do you think is out across all our sites? 70, 20, 40, okay, if we split those two, about 30% is kelp. Then we get this here, which is algal turf and algal slime. It's like just tufty, slimy algal stuff. And then you get this red stuff, which is calcareous encrusting algae. It's red algae. That's this red. So nearly half the cover is stuff that has no structural benefit for anything. Stuff, you know, nothing can really live in it. And often that's the urchin barren. Right? Urchin barrens typically are the red encrusting algae and turf and a bit of slime. So you might have, I can't tell you barrens here, but you might have more barrens than kelp, for example. So even though 32 is pretty good, you've got potentially a lot of urchin barons, and so that's something to keep an eye on. Okay, if they start growing, then you're going to see what's happening in other places where you start to lose your kelp. Okay, so now some interesting sightings. So I've got 2 more sections to go through. I'm going to show you some species that we've recorded in the bay that I think are interesting because they shouldn't be here, or maybe we're at the extent of their range, so they don't go any further north or south. And then I'm going to talk about some of my personal favourites. I just have some that if I see them here, I get pretty excited about. I love to see them. So let's just I suppose it's a bit like, meet some of the more interesting things that we see when we're out doing surveys. Now, the first interesting sighting I want to talk about is a fish that eats coral and is tropical. What's it doing here? How much coral do you have? So it's the 3-band butterfly fish. This is it here. This is not a Jervis Bay photograph. This is coral reef. This is, I think, Lord Howe Island. So this is all coral here, all these plate corals. They live in the tropics. They eat coral, and we've recorded them on 2 years here. How come? Well, just in Finding Nemo, you've got the East Australian current bringing larvae down, little fish then hatch, and they say, I've got to live on a reef, I've got to live on a reef, and they swim into Jervis Bay. They go, there's no coral here, what am I going to eat? So they might just be arriving, maybe they didn't even survive the winter, but we did record them in 2 years. So that was an interesting one. When I looked through the data, I said, what's that doing here? And you might say, well, hang on, it might have been a mistake. All of these unusual sightings we verify with a photograph. Okay, so if I tried to say I saw this tropical species here on this trip, the first thing RLS will say to me is, you've got a photo, John? Because if you don't, we're not putting it in the data set. So these are all verified by experts checking the species IDs. So we've had that corallivore, a fish that eats coral, on 2 years. We've also had the inscribed wrasse on 2 years, 2000 and 2003. I've only seen these in places like Lord Howe and Norfolk Island. So if I saw one here, I go, what's it doing here? Probably the similar story to the 3-band butterfly fish. It's just nice to think that we've got larvae travelling down. They'd probably get washed out to sea and die, but they might, if they're lucky, get into the bay, live a little while, at least party for a short while before getting eaten. So that's the inscribed wrasse. This one here, the black saddle toby. This fish is Africa, Asia, tropical Pacific, and on 2 occasions found its way into Jervis Bay. So you see a bit of a pattern there. We're getting some tropicals on the odd occasion. Now with climate change, there is the expectation that that will increase and you get more tropical visitors getting carried further south as the water warms, and the EAC is actually getting stronger as well. So the water volume is increasing and it's getting warmer. So you might find more tropical visitors in future years. With the invertebrates, it was interesting when I looked at the invertebrates, it's more coming the other way. So with the invertebrates, what we're finding is species that this is as far north as they go. So this is the one that really stood out to me. It's the southern biscuit star. It's been recorded 3 times just in 1 year. So that was probably a recruitment event where some larvae just got carried up, maybe on a, the East Australian current has some gyres and some eddies. So maybe it kicked up some larvae and they got into the bay. But that's as far north as they go. So it's a temperate visitor coming into Jervis Bay. Get the idea? It's a bit like a bus station here, right? With the odd tropical bus coming through and the odd temperate bus coming through. And same with this pheasant shell, which is also a temperate species down around Victoria and Tassie and so on, and found its way up here. So they're exciting for us when we go and do surveys because we find something that's out of place. And we photograph it and we show, and so then it becomes a record. Okay, that's never been recorded in Jervis Bay before. So we as surveyors are interested in those sorts of fish. Now I'm going to show you a few that I personally really like. And then we'll open up for questions. This is the title, some of my favourites, but actually I've put one of my favourites in the slide, and that is the giant cuttlefish, Sepia apama, because cuttlefish are so smart. You look them in the eye, you can tell. You can tell they're thinking this, they're thinking that, and also their ability to change colour and texture. They can change the texture of their back from rough to smooth. They can make stripes go down their back like a neon sign, like an animated neon sign. So they're incredible animals to encounter, and they're big. You know, the carapace of a giant cuttle can be 1/2 metre long. That's just not the tentacles, that's just the carapace. So that's one of my favourites. But my next favourite is also a really smart animal. Some think these are the smartest invertebrates on the planet. The octopus. It's called a gloomy octopus, which isn't a great name, but once again, they are smart animals, you can tell. Often if you find one in its hole, you sort of wriggle your finger in the sand and it sends out a tentacle to explore what you're doing. You can see once again it's interacting, it's thinking. This is a photograph from the Octopolis, which is that area where there's just, I don't know how many octopus do you reckon are Octopolis? Dozens or more, maybe lots, yeah. So you just look out and there are all these octopus mounds with all these octopus like poking their eyes out of them. This is one here is sort of, you know, spreading himself or herself over this mound. And I think it's probably because the area has lots of these cockles in it. So they're eating those and so making their little octopus gardens. Yeah, it's a boat. My next one, I just love whenever I encounter one of these because their power and grace and the electric energy that you can feel in the water just from the power of the animal. And also the East Coast population is critically endangered. So to see one of these is really special, and that's of course the grey nurse shark. I've seen several of these on surveys here, a couple of places where I've seen them more than once. But encountering one of these, and they never threaten, they just sort of go past you, look at you, go, say, okay, another one of those things that blows bubbles. But they are amazing. It'd be like going to Africa and, you know, a lion just walking past and looking at you going, yeah, okay, whatever, off you go. So it's great to see these. It's great to see them both because they're critically endangered, and so it's special to see one, but also just to think, this is an apex predator. I'm sitting here and there it goes past me. The next one is one of my favourites because these guys or gals have attitude. The wobbegong, this is a banded wobbegong. And this one swam in, I was doing my survey and the tape's there and it swam in and they swim along and then they settle down like a hovercraft. So he swims in, settles down right on my transect tape. And I'm doing my recording on, you better move by the time I get back. I'm coming back, still there. Now I've got to reel in. Reeling in. Luckily it was from that direction, not this direction, the sharp end. So I came back and I'm, get off my tape. So wobbegongs are amazing. They're big. We got one today on transect, it was 2 metres long, head this wide. And I know it was 2 metres long because my slate's 35 centimetres and I measured 6 of them. So I didn't actually hold it right against the animal, but I held it up enough, as close as I was prepared to go. So I love wobbegongs. And also the biggest I've ever seen, it's hard to see because the resolution not great. That's a stingray. And here is Sue, I think, and there's Sue sort of there. Now, okay, so you got the camera effect, but still, this was a massive stingray. When the early days of Europeans entering Botany Bay, they recorded these around 300 kilos. So I don't know if this one was that big, but it was big. It was like probably weighed 2 of me at least. So amazing to see those. That was, I think that was off Bowen Island. Does that sound right? Yeah, I haven't surveyed Murray's much, so it was not near shore. It wasn't like hanging around the boat or anything. It was out, so it would have been off Bowen Island. Another one of my favourites, and that's because some of us have been monitoring these up and down the east coast for a few years now, and they're always amazing to see because on Earth there actually are real dragons. And we've got them here, weedy sea dragon. This photograph would have been taken off Bowen Island as well. And beautiful animals, just like a dragon, long snout, breathes fire, but gets immediately extinguished. That was a joke. Thanks for those of you who, dad jokes, I know. Still. So I love seeing these. This is a male because it's a little bit thinner in the body here, and amazing to see and watch them. Of course, the males are the ones that get pregnant, so the ones that carry the eggs and look after them under their tail. The female makes the eggs but gives them to the male to look after, so basically he has the pregnancy, if you like. We hear about it, yeah. Another one of my personal favourites is Port Jackson shark, but I really love the babies because they are so cute. They are so cute. They're this big, and they just, and when you go up to them, they don't swim very far. They just think, I'm going to swim away from you, and they go like there. Okay, if I was a predator, that's not going to save you. So these are amazing to encounter, and sometimes you get them in sort of depressions where you get like several of them in a bit of a group, so that's amazing to encounter. Another encounter which I love, if ever I see one, is the cauliflower or the pink soft coral. Now this soft coral, Dendronaphthia australis, is really special because it only lives pretty much from Jervis Bay to Port Stephens. Nowhere else in the world. That's the only place you'll ever find one. And if that population disappears, that's it. So we've got this amazing soft coral that's local, that's ours, and it inflates itself to get up to feed. So this single polyp here, which makes the body, is just pumped up through hydraulic pressure. And so it gets up and it can feed, and then when the current stops, it contracts back down again. So incredible. Well, the biggest ones I've seen of these, not in Jervis Bay, but in Sydney, you couldn't get your arms around. That's one colony. And there are fields of them. Really something. So yeah, amazing to see these. I've got one more, and then a couple of slides, and we wrap up for questions. I've got to finish on one of my favourites, just because it's the weirdest fish, I reckon. And that's because it lives its life upside down. I call it the upside down fish. Hyperplectros annulatus, for those of you who care about that. But it lives on the underside of overhangs. Yeah, yeah. No, but these days, when you do that, the camera corrects itself. I've tried that. So I love these because they're not that common to see because they live in caves, and so unless you stick your head in a cave, which we do for Method 2, you won't find them. But yeah, and they swim, but they swim upside down like they're like anti-gravity or something. So the upside down fish. So a couple more slides just to sort of wrap this up. Now, you might say, wow, that's all amazing. It's nice to see. You can go and look at all this yourself. So Reef Life Survey, because we gather our data through volunteers, all of our information is available online. Okay? So all these photographs, these are my photographs, except for the ones that said this is not your... Oh, no, actually, they're mine too. But a lot of these are in the Reef Life Survey library, and much better photos than these taken by better photographers than me. So you can go online, you can say, I'm interested in, don't say upside down fish, it won't recognise that, but if you said that, most of the time you can look up the common name, you can look at its distribution or photographs of it, you can look at other fish that people mix this fish up with, so that's really helpful for checking IDs. I thought it was this and it's actually that. So these are some of the pages from the Reef Life Survey website. You can see, maps of the sites where this particular fish is found. You can also draw a box around Jervis Bay and say, show me everything that lives in Jervis Bay. Sorted by the most frequent to the least frequent. That's an amazing thing. If you go for a snorkel and you say, I don't know that fish, go into what we call the Frequency Explorer, because it's based on the frequency of sightings. draw a box around Jervis, and if it's a common one, it'll be near the top, and you just scroll down. Some other really good things about the Frequency Explorer, this view isn't, but there's a view where you get multiple photos of the same animal, and you're like, okay, why? A lot of species change as they grow. So the multi-photo view is really helpful for identifying juveniles, for example. And there's also this view here, the Reef Life Explorer. You know those indicators I showed you, the biomass and so on? You can look at those yourself too for Jervis Bay. I think the data set's got Jervis Bay in it now. As each area gets enough data, we turn it on. So I think Jervis Bay is turned on. At the moment, this view is showing Eastern and Central Australia, but still, you can go in and say, show me the biomass trends, show me whatever yourself. So my final slide before questions is thank you, first of all, for giving the time, but how could you get involved? Well, if you're a diver and you're mad like some of us, you could start doing surveys and we'd train you up and off you go. But there are lots of other things you can do. First of all, you can visit our website and use it. Like any website, the more people use it, the more it gets picked up and other people find it. Second, you can join our Facebook page or follow us on Twitter or whatever and share our posts. So that we do this to get the word out. The reason I give my time is so that other people can see what I think is an amazing thing, the underwater world. So the more you share things and tell others and so on, the more that inspires me to go out and do surveys. You can donate if you want to support us financially. We're always looking for funds, and that money goes into supporting trips like this. And if you want to get involved as a volunteer and do surveys and so on, you can go on and register interest, and then we start the conversation with you about, okay, how much diving have you done, and so on. So that wraps up my presentation. You've been a great audience listening to all of this.